All right, let's look at chapter 14. So chapter 14 deals with gene expression. And gene expression is the expression of a gene, or another way to look at it is we're going to look at this. We're going to look at how we go from DNA to making a protein. So we're going to go from DNA to RNA to protein, and from the protein we're going to make a trait. Now in this we're going to look at a gene. And a gene is a sequence of DNA nucleotides that codes for a protein. All right? Now the gene does not directly affect the phenotype. The gene product affects the phenotype. So if we make a mistake in any of these processes here, that's going to affect the expression of that trait. So let's look at RNA very briefly. Now I don't have a picture of RNA, but RNA stands for ribonucleic acid. And RNA is going to be what makes the proteins. Now RNA is a large molecule composed of nucleotides. All right, if we look at RNA nucleotide, it is made of a phosphate group, a ribose sugar, and a nitrogenous base. And once again, there are 4 nitrogenous bases. So there's adenine, guanine, cytosine, and instead of thymine, which we see in DNA, we use uracil, which is made of RNA, in RNA. Okay? RNA is also a single-stranded molecule. There's no helix, and there are 3 types that are made. Now, let's take a look at the main steps here. If we're looking at a prokaryotic cell, there are 2 processes, transcription and then translation. When you look at a eukaryotic cell, there are 3 steps here, essentially. There's transcription, there's an RNA processing, which we'll call modification of RNA, and then translation. All right? So let's take a look at this process here. This shows at a higher magnification here. So we're going to go from DNA to RNA, and from RNA, we're going to make that protein, which is made of amino acids. All right? So let's take a look at transcription. So transcription is the synthesis of an RNA on a DNA template. All right? It is the first step in gene expression. This occurs in the nucleus in eukaryotic cells, and it makes all forms of RNA. But for the purposes of transcription, we're just looking at making what is known as messenger RNA, or mRNA. And what mRNA is essentially a copy of the gene. All right? So the first step in transcription is what is known as chain initiation. And so this begins when RNA polymerase attaches to a promoter. So this is trying to show RNA polymerase, and the green is trying to show the promoter. RNA polymerase is a protein is an enzyme that makes RNA. And a promoter is a specific DNA sequence that marks the beginning of a gene. It defines the start of the gene. It also tells the direction of transcription. So in this case, it's left to right instead of right to left. All right? It also tells us which strand is to be copied. Because if you go in any other directions there, you're going to make a different messenger RNA transcript. All right, so that's chain initiation. So let's look at chain elongation. So chain elongation is the next step, and this is where the DNA unwinds slightly, what we see there. And transcription is going to occur by complementary base pairing. All right, so if we go to the next picture here, right, so this shows us this small unwinding of DNA, and then new nucleotides are put into position here, all right, by complementary base pairing. And so A goes with T, C goes with G, right? But here you see an A on DNA, but now instead of having a T, because it is RNA, we're going to use a U instead, all right? And this process will continue down the gene, right? And as we move, the RNA polymerase moves down the gene, new complementary base pairs are put into position, and our messenger RNA transcript elongates. And then we get to the end here, and at the end it's called chain termination. And so here, RNA polymerase hits a terminator, all right? And so the terminator is a specific sequence of DNA located that marks the end of a gene, all right? And so that ends transcription, and that releases the messenger RNA transcript, all right? But when we're talking about a eukaryotic cell, we're not done. So we go to the next step, which is known as modification of the messenger RNA. So in modification of messenger RNA, here it is, here's our gene, here's our messenger RNA transcript that we transcribed. And what we're going to do is we're going to put a cap on it and a tail. Now the cap is one guanine, the tail is several adenines. And so what this does is it helps protect the messenger RNA against cellular enzymes, but what's probably more important than that is that it allows the ribosomes to recognize the messenger RNA transcript. Because all the messenger RNA transcripts have the same cap and tail, well, that makes it easier for the ribosomes to recognize those. All right? So the next thing that's going to happen is removal of introns. So introns are non-coding regions of DNA. So you can see them up here. So an intron, so this part of DNA is not used in making a protein. All right? It's still made of adenine, guanine, cytosine, and thymine. But that part is not used in making a protein. All right? Next are exons, and so exons are shown in the darker blue. So exons are coding regions of DNA. So those are parts of the DNA that are used in making a protein. So what we do is we transcribe the entire gene, both the introns and exons, and then what we do is we cut out those introns and we put those exons together, making a mature messenger RNA transcript, and that can now leave the nucleus and go to a ribosome. All right? Now, if you guys remember, last time, so back in chapter 13, I talked about how every time we make a copy of our DNA, we're going to make 3 errors. If those errors occur in an exon, well, that can be bad. But if it occurs on an intron, since that intron is not used in making a protein, we still make the same proteins, so there's no harm that occurs when they occur there.